Assalamu alaikum. Today we are going to start another lecture that is on control of gene expression in neurons. How many new genes are there in the human beings? First of all, we answer this question, that is 30,000 genes are present in the human genome. It lasts a third of our 30,000 genes that make the human genome are active or expressed in the brain. So we can say that 30,000 genes are present in the human genome. This is the highest proportion of the genes expressed in any part of the body. These genes influence the development and function of the brain, ultimately control how we move, think and feel and behave. Combined with the effects of the environment changes in these cases can also determine whether we are at risk for a particular disease, and if we are, the course it might follow. So genes are basically for the our whole body control. So certain genes make proteins that in turn make neurotransmitters. Some genes work for the neurotransmitters, some genes for the other functions like transfer of the stimulation of the stimulus after getting the message from the effector and sensory or motor neurons. So these transmitters work for the signaling. Other proteins are important for establishing synapses or for disposing of excess neurotransmitters like comet genes. So, the comet gene provides instructions for making an enzyme and catechol or methyl transferase. Comet, what is the comet? Comet are the enzymes, that is catechol or methyl transferase. These are for the instruction of making the enzyme. Okay, ये provide करते हैं information. Two versions of enzymes are made from the genes. There are two versions of enzymes for the to make the genes: the longer form and the shorter form. Longer form called membrane bound catechol or methyl transferase, that is MB-COMT, is chiefly produced by the nerve cells in the brain. Everyone carries two copies of the comet gene, one inherited from each parent. One is inherited from the each parent. The study showed the people with two copies of the MET form of the comet gene had a much more pronounced response to the pain than those who carried two copies of the well from the gene. Okay, two forms. There is one form is comet and the other form is MET. Well, that is for the making of the genes. Like these are the enzymes which function for the making of the genes. Like for the pain, that is from the carried out from the well gene. Okay, gene expression. How gene expression occurs? Gene expression involves a series of steps in which the information in the sequence of bases in DNA specifies makeup of the cell's protein. The proteins affect the phenotype from readily. Okay, gene expression involve in the basic steps, like a sequence of the bases in the DNA specifies the cell's protein. So the protein which affects the phenotype. So protein affect the phenotype from readily observable physical traits. The subtle changes detectable only at the biochemical level. So the protein affect the phenotype. Phenotype is the thing that is visible from visibly we can see it, that is phenotype. From readily observable physical traits, that are physical traits. From phenotype is basically physical traits that can be observable to subtle changes detectable only by the biochemical level. Then it convert into the biochemical level, that is a chemical nature. The first major step of the gene expression is transcription, the synthesis of RNA molecules complementary to the DNA. And the second is translation. Gene expression involves, first step is transcription and second one is translation, in which RNA becomes coded template to direct polypeptide synthesis. RNA will convert into the polypeptide synthesis, but in transcription, RNA molecules are complementary to the DNA. How genes and proteins are connected? This is the basic question. We have to find it on next later stages. Classification, logical answer to this question. Okay, classification of gene with respect to their expression: constitutive or controllable genes. Constitutive are the housekeeping genes, are expressed at a fixed rate irrespective of the cell condition. Their structure is simpler. Constitutive or housekeeping are simpler one and fixed in rate, and they are expressed in a fixed rate, but in a controllable genes are expressed only when needed, like their amount may increase or decrease with respect to their basal level in different conditions. Their structure is relatively complicated with some response elements. Okay, different cell types produce different sets of proteins, the extent of the differences in gene expression between different cell types may be roughly gauged by comparing the protein composition of cells in a liver, brain, and so on, using the technique of two-dimensional gel electrophoresis. The extent of differences in gene expression between cell types may be roughly gauged by comparing the protein composition. It is checked by the protein composition, in the liver, in the brain. Also, using two-dimensional techniques, we can know about this. Okay, experiments of this kind reveal that the proteins are common to the cells of the multicellular organisms, as they are complex and protein is in a higher amounts, you can say. These housekeeping are simpler and their rate is fixed. Okay, include the structural proteins of the chromosomes, DNA or RNA polymerase, and these are all enzymes, DNA repair enzymes, ribosomal protein enzymes involved in glycolysis. These are the life cycles processes and other basic metabolic processes, and many of proteins that form the cytoskeleton. Each different of the cell type also produces specialized proteins that are responsible for the distinctive properties. So the proteins are for the different functions, different cycles for the different various activities. Like gene regulation. What is gene regulation? Virtually every cell in your body contains a complete set of genes. There are complete set of genes in your body, but they are not all turned on in every station. Each cell in your body expresses only a small subset of genes at any time. During development, different cells express different sets of genes in a precisely regulated manner. So, gene regulation occurs at the level of the transcription or the production of a mRNA. Transcription is the production of mRNA. So gene regulation may occur at that time. A given cell transcribes only a specific set of genes and not others. Genetic information always goes from DNA to RNA, then to the protein. That is the steps. That is also called central dogma. Like transcription, then translation occurs. That is called gene regulation. Okay, every cell has same DNA and others, therefore the same genes, but different genes need to be on or off in different types of cells. Therefore, gene expression must be regulated, like a embryo cell, in a bone cell, in a liver cell, muscle cell, or sperm cell. Okay, now the next one is thing. Gene expression must be regulated in several different dimensions, like how they change in a 10 week or in a 14 weeks, you can say it in a 1 day or 6 months. Different stages gene may be turned on, their activity may promote the activity, or their regulation may change the activity to lead to the different dimensions. The development and function of the CNS requires accurate gene transcription control in a response to proper environmental signals. For the transcription, that's the main process for the central dogma. For the translation to the to leads to the translation. Epigenetic mechanism include DNA methylation, histone modifications, and other concomitant remodeling events are critically important in mediating precise neural gene regulation. So these are the steps important for the gene regulation. Next thing is, one of the striking features of the neuron is their intense utilization of RNA, regulatory pathways of perform, and diverse cellular functions such as axon guidance and synaptic plasticity. That was all about, and we will discuss in detail in a later video.